Welcome back, class. We'll continue our conversation on the tools of discovery, having our head examined part 3. There are a variety of ways in which we can try to look into the brain to see what's actually going on. In the previous lecture segment, we talked about a variety of techniques and devices such as the EEG, MEG, PET scans, MRI, and fMRI. Well, there are other new activities as well that are being conducted to try to find out not only about how we can image the brain in new ways, but also how we can understand it via new activities such as neuromarketing and neurolaw and neuropolitics, which we'll talk about in a second. The Human Connectome Project is using cutting-edge methods to map the brain's interconnected networks of neurons. Scientists create a multicolored symphony of neural fibers that transport water through different brain regions. By this way, we're getting another view, another look at how the brain is connected and how it actually works, and hopefully giving us better understanding of this wonderful thing that we have that has really catapulted us to the top of the food chain, so to speak, our brain. So, what is all this neuro stuff, this notion of neuromarketing, neurolaw, and things of that nature? Well, just to give you a few examples, there is a video that's available to you on your Canvas site, by the way. We have neuropolitics, is a science which investigates the interplay between the brain and politics. We'll be seeing a lot more of that in play over the next few months. Neurolaw is a field of interdisciplinary study that explores the effects of discoveries in neuroscience on legal rules and standards. And then I think that's the one that the video will be shown in your Canvas site. It's going to be on neurolaw. Then we have neuromarketing. It's a commercial marketing communication field that applies neuropsychology to marketing research, studying consumers' sensory-motor, cognitive, and affective, that is, emotional responses to marketing stimuli. Basically, what it comes down to, we're trying to understand the brain slash mind in so many ways to try to help us when it comes to things like selecting political candidates or getting someone the right sentence for a jury trial, for example, that may have some sort of neuroanatomy or some other issue dealing with psychology involved, or neuromarketing, trying to make sure that if you are a company, that your product and your services are being optimized emotionally in the eyes of your potential customers. Now, let's talk about this thing, the brain. The brain is a wonderful thing, but there are several versions of the brain in a way. There are several levels of brain. What we're going to talk about now is what we refer to as our old brain, or the older brain structures. The older brain structures function without any conscious efforts. We can imagine that these were some of the first parts of our brain that was developed, one of the first ones, the ones which were critical to our survival before we developed later on the part of the brain which is the cerebral cortex, which really allows us to put ourselves way above most other animals in the world. So the older brain structures function without any conscious efforts on our part. We have the brainstem, the oldest part in central brain core, automatic survival functions, crossover point. And what we mean by the crossover point, you may be familiar with the idea that when we control our left arm, it's our right hemisphere of our brain that controls our left part of our body. And then the right part of our brain, the right brain, controls the left part of our body, and the left brain controls the right part of our body. So the idea is this crossover point. This crossover point happens really low in the brain at the area of the brain stem. So when someone has damage to their left cerebral hemisphere, usually if they have a stroke, for example, you'll see the results on the right side of their face and vice versa. So the brainstem, one of the oldest structures in the brain. Then we have the medulla. This is the brainstem base, and this area seems to control our heartbeat and our breathing. Okay, our heartbeat and our breathing. So the Other structures of the brain are going to be things like the thalamus, the brain's sensory control center, located on the top of the brainstem. It directs messages to the sensory receiving areas and the cortex and transmits replies to the cerebellum and the medulla. The reticular formation, nerve network that travels through the brainstem and to the thalamus and plays an important role in movement, output, and balance, and enabling nonverbal learning and memory. So the thalamus, central control center at the top of the brainstem, it directs messages to the cortex sensory receiving areas. The reticular formation, a nerve network that travels through the brainstem and on the thalamus, and it controls our level of arousal. When they do experiments on animals, if they damage or lesion, as they refer to it sometimes, the area of the reticular formation, they can put the animal to be always alert and aroused, or put the animal basically to in a comatose type of state. The cerebellum, also referred to as the little brain, because it looks like a little miniature brain, is at the brainstem rear. It processes sensory input, coordinates voluntary movement, output, and balance, and enables nonverbal learning and memory. A couple of other things about the cerebellum. The cerebellum is also a key factor when you are going through a sobriety test. When someone drinks alcohol a little bit too much and they're out and about on the road driving, the cops pull them over. They think they may have been drinking. They give them a sobriety test. Take your index finger and touch the tip of your nose, or walk a straight line. They're doing that because one of the first things that happens when you've had a little bit too much to drink, your cerebellum will be affected. And see what we see here, the cerebellum coordinates voluntary movement output. And so they're testing to see if your cerebellum is functioning properly. If it's not functioning properly, it's due most likely to something that you have ingested, alcohol, for example, or some other type of drug. That is why they do that particular test. It's testing basically the cerebellum. Now we have the limbic system. The limbic system, located below the cerebral hemispheres, and it regulates emotions and drives. So in the limbic system, the neural system, including the amygdala, the hypothalamus, and the hippocampus, located below the cerebral hemispheres, associated with emotions and drives. The amygdala is two lima bean-sized neural clusters in the limbic system, and they're linked to emotion. When they have people in the fMRI machines, and they show them something that is an emotionally arousing stimuli, the amygdala will usually light up, meaning that it's processing information. So that's the part of the brain that's being utilized at that time. The hypothalamus is a neural structure lying between the thalamus, below the thalamus. It directs several maintenance activities such as eating, drinking, and body temperature. It helps govern the endocrine system, which we saw a little bit earlier, via the pituitary gland, and is linked to emotion and reward. And then we have the hippocampus, a neural center located in the limbic system. It helps process explicit, conscious memories of facts and events for storage. So the amygdala, this is a graph here, an image of it. We can see the amygdala, again, the two bean-sized neural clusters for emotions, the hypothalamus primarily for emotion and reward and maintenance activities, and the hippocampus, the seahorse-shaped neural structure for explicit conscious memory. A couple of things real quickly. I already told you about the amygdala, that it will light up if you're seeing something that's very emotional. We also want to see that the hippocampus is where information is going to be present before it is stored for more long-term storage. So when we have damage to the hippocampus, then it's going to be very difficult to get information that you are learning at the moment and utilizing short-term memory, and we'll talk more about memory later, and be able to shuttle that information to long-term memory storage. So hippocampus damage will have an impact on your memory, particularly long-term memory. You won't be able to place things into long-term memory storage. This graph here, this figure, shows a lot of the things that we will talk about, as well as some of the things that we've already talked about. It shows you where they're basically located in structures in relation to one another. Be familiar with it. That'll be a good thing to do. A couple of things I want to point out real quickly here. The far right, we have the cerebral cortex, and we'll be talking about that real soon. The cerebral cortex is the ultimate control and information processing center. Normally, when we think of brain, we are thinking about the cerebral cortex, that wrinkly thing that we associate with the brain. That's it. Okay. Now there are other structures, like we've talked about the older structures, but the newest structure, by way of our development as a species, is that wrinkly brain, that cerebral cortex. Question to you: why is the brain wrinkled? Why isn't it smooth? Well, the logical answer is this. The brain is wrinkled because you're able to get more space, more brain in a smaller container. Basically, if our brain was smooth, our heads would have to be ginormous. It would have to be really huge heads, which would require huge necks, huge shoulders, and it becomes a whole thing. Therefore, the way we were developed, our brains are wrinkled so we can get more brain space and a smaller container, a smaller skull. So we have the corpus callosum, which is the fibers that connect the two cerebral hemispheres, and we'll see more about that in a second. But I just want to give you an idea of all these brain structures that we have talked about, and we will talk about, and you'll read about as well. Okay, so let's get into some of the nitty-gritty of this brain, particularly the new brain. We've talked about some of the old brain structures. Now let's talk about the big guy, the cerebral cortex, the ultimate control and information processing center of the body. 20 to 23 billion brain nerve cells and 300 trillion synaptic connections. So there's a lot of stuff going on in there. The cerebral cortex, the wrinkled parts of the wrinkled brain. The cerebral hemispheres, paired and covered with intricate fabric of interconnected neural cells. The cortex of each hemisphere is divided into 4 lobes separated by fissures, or little, sort of like little canyons in the brain. So here's some of the terms you want to be familiar with, and what are some of the names of the lobes and where they're located in the head. So we have frontal lobes, parietal lobes, occipital lobes, and temporal lobes. All right, the frontal lobes, portion of the cerebral cortex lying just behind the forehead, involved in speaking and muscle movements and making plans and judgments. The parietal lobe, portion of the cerebral cortex lying at the top of the head and toward the rear, receives sensory input for touch and body position. The occipital lobe, portion of the cerebral cortex lying at the back of the head, includes areas that receive information from the visual fields. And then the temporal lobe, portion of the cerebral cortex lying roughly above the ears, includes the auditory areas, each of which receives information primarily from the opposite ear. Again, remember we had that crossover at the brainstem that we talked about earlier. So that means that things on your left side are normally going to be controlled by the opposite side brain. Now let's look at the other image we have here on this slide. The brain has a left and right hemisphere, and the corpus callosum is the bridge that sort of connects those two together. Okay, so that's the layout of the cerebral cortex, our brain. 2 lobes, left and right hemisphere, 2 halves, right and left hemisphere, and 4 lobes, frontal, parietal, occipital, and temporal, each with certain functionality to allow us to survive in this world. So let's look at motor functions. The motor cortex, the rear of frontal lobe, controls voluntary movements. Mapping the motor cortex, the body areas requiring precise control occupy the most cortical space. Get this idea in your mind. that when you are processing information, the more information you take in, the more space required to process it. So things which are very sensitive on your body will require a great deal of cortical space to process all that information. Things that are not very sensitive in your body will require less space on the brain to process that information. And then we have brain-machine interfaces. Why do we want to study all this stuff anyway? Well, a lot of it has to do with what we can do when we know how the brain actually functions. How does the brain talk and communicate to your fingers to tell them to flex or to splay or to point? If we know how that's done, then we have a possibility of being able to mimic that function with prosthetics, for example. So brain-machine interfaces rely a great deal on how we actually work when we have all our body parts. Figure that out, then we can maybe figure out how to hook a artificial arm onto somebody in such a way that they can use their already designed brain parts to try to control it. So device implants are understudied to detect and facilitate motor cortex activities in humans. Implant recipients have learned to direct robotic arms with their thoughts. And the idea is that as our understanding of the brain matures and develops and grows, maybe our ability will grow as well in the area of prosthetics to be able to build a robotic arm and legs and things of that nature, so that people who may have lost such things will be able to have some semblance of a life with those functionalities available to them again. Here's the graph that we were talking about a little bit earlier with the functions of the cortex, and we can see here that the size that we see depends on the sensitivity required of a certain body part or the sensitivity of the control. So basically the same idea. The more control, the more sensitive something is, the more processing and more space in the brain that it'll take up. Sensory functions. Somatosensory cortex receives incoming messages from the skin, senses, and movement of body parts in front of the parietal lobes, parallel to and behind the motor cortex. Body region sensitivity equals larger somatosensory cortex. Body region sensitivity equals larger somatosensory cortex. The visual cortex receives input from eyes, sends information to other task specialized areas. We do have very specific areas of the brain that are designed to do some very amazing things. For example, face identification. We have specialized cells, brain cells, that are designed to allow us to quickly identify faces, to detect emotion, to recognize faces. And believe it or not, there's such a situation where you can have damage to a portion of your brain. You can see just fine, but your ability to recognize faces will be impaired. No matter what you see, who you see, you will not be able to recognize their face, although your vision will be perfect. 2020, you can see everything, but your ability to recognize, to perceive, would not be there. Again, remember as we talked about in one of the first lecture segments about sensation and perception. Those are two different things. Sensation is you just sensing stuff, and perception is how you make sense about what you're seeing. We'll talk more about that in a future chapter as well. So let's look at the sensory functions of the auditory cortex. It receives information from ears. It takes a very circuitous route from one ear to the auditory receiving area above the other ear in the temporal lobes. Here you can see where it's located. And the green area is the visual cortex. So we have identified these areas of the brain that seems to specialize in certain functions to process certain types of sensory information.